With the mind map on TCA cycle is very simple. So right now, think of the whole pathway. So pyruvate, you get acetyl CoA A. Acetyl CoA A joins with um, oxaloacetate via the enzyme citrate synthase, and then you get citrate, and from citrate to isocitrate to alpha ketoglutarate, and from there to succinyl CoA, and then to succinate to fumarate, and finally to malate. Right, so the key enzymes involved, from pyruvate to acetyl CoA, pyruvate dehydrogenase is involved over there, where um, NADH is reproduced, and then from citrate to isocitrate, aconitase enzyme is implicated over here, and from isocitrate to alpha ketoglutarate, you have isocitrate dehydrogenase, and then from alpha ketoglutarate um, to succinyl CoA, you have alpha ketoglutarate dehydrogenase, succinyl CoA to succinate succinyl CoA synthase, and then from succinate to fumarate, you have succinate dehydrogenase, and from fumarate to malate, you have fumarase, and then from fumarase to malate to oxalo to malate, you have from fumarate to malate, you have fumarase, but from malate to oxaloacetate, you have malate dehydrogenase. All right, so basically that's it. So at the beginning, you have from isocitrate to alpha ketoglutarate, NADH and CO2 are being produced. Another NADH from alpha ketoglutarate to succinyl CoA, and also FADH2 from succinate to fumarate, and another NADH from malate to oxaloacetate. Right, so those are the 8 steps that are being involved in the citric acid cycle, or the tricarboxylic acid cycle, Krebs cycle. Right, so note that glycolysis takes place in the cytosol, fatty acid oxidation in the mitochondrial matrix, the same as citric acid cycle, but for the for oxidative phosphorylation occurs in the inner membrane of the mitochondria. So that's basically it. With the biomedical importance, the citric acid is the final common pathway of the aerobic oxidation of carbohydrates, lipid, and protein, because glucose, fatty acids, and most amino acids are metabolized to acetyl CoA or intermediates of the cycle, and it also has a role, a central role in gluconeogenesis, lipogenesis, and intervention of interconversion of amino acids. So, what are the functions of the Krebs cycle? It has catabolic and anabolic functions. So, with the it has energy generation as a function, like I said, reducing equivalent NADH, FADH2, GTP is being produced, and provides carbon dioxide for gluconeogenesis, fatty acid synthesis, urea synthesis, and nucleotide synthesis, and also provides precursors for gluconeogenesis, all the intermediates, and amino acid synthesis, the non-essential amino acids, heme synthesis, like succinyl CoA, and fatty acid synthesis, that's the citrate, and it regulates other pathways, like the citrate inhibits phosphofructokinase. The TCA cycle, like I said, the tricarboxylic acid or Krebs cycle, citric acid cycle, functions to burn acetyl CoA made from fat, glucose, or protein in order to make ATP in cooperation with oxidative phosphorylation. Right, so all the cells in the mitochondria TCA cycle could occur. and with the ATP yields with pyruvate, say 15 ATPs, acetyl CoA 12. You know, four ATPs are produced from glycolysis, but only two, um, and two is being utilized in the process. So only two is the net, or from pyruvate to acetyl CoA, and all the processes happening. You can think of 15 ATPs and 12 ATPs too for acetyl CoA. Alright, so it has 8 steps like I talked about, and it requires 8 enzymes, and they are located in the mitochondrial matrix, and the final product is oxaloacetate. So 3 NADH molecules are produced, 1 GTP molecule is produced, 1 ADH2 molecules also being produced, and the cycle is accompanied by the liberation of 2 carbon dioxide molecules. So the cycle itself neither generates ATP nor includes oxygen as a reactant, but it removes electrons from acetyl CoA and uses them to form NADH and FADH2, which are high energy electron carriers, and these reducing equivalents are transferred to the respiratory chain, where rate oxidation of each NADH results in the formation of 3 ATP, and rate oxidation of FADH2 in formation of 2 ATPs. So after glycolysis, what next? Remember, for glycolysis, 2 pyruvates, 2 ATPs, 2 NADH, 2 hydrogen ions, 2 H2O. So the cells need to regenerate the NAD plus that was utilized in glycolysis, and NAD plus regeneration can be accomplished by aerobic or anaerobic metabolism. So the 3 possible fates of pyruvates could be aerobic metabolism, that is where you have your acetyl CoA to the tricarboxylic acid cycle to electron transport, or from pyruvate to L-lactate, that is homolactic fermentation or anaerobic metabolism. And the NADH plus hydrogen ions is being utilized by lactate dehydrogenase to form NAD plus, and then from the pyruvates to anaerobic metabolism to get acetaldehyde, where acetaldehyde is converted to ethanol by alcohol dehydrogenase utilizing NADH and hydrogen ions form NAD plus. So that's the alcohol fermentation. So glycolysis does not require oxygen from the air, but it does require the oxidant NAD plus. And lactic acid fermentation occurs in several types of animals and microorganisms. Ethanol fermentation is usually in yeast and other microorganisms. So there are several, there are different coenzymes or prosthetic groups required for pyruvate dehydrogenase complex, and they are thymine pyrophosphate, flavinadenine dinucleotide, coenzyme A, nicotinamide adenine dinucleotide, that's the NAD plus, and then lipoids. Yeah, so thymine. So there are 4 different vitamins required in human nutritional, very vital components of this particular system. As you see, thymine in CPP as a thymine pyrophosphate, and flavin in FAD, that is flavin adenine dinucleotide, and niacin in NAD, nicotinamide adenine dinucleotide, and pantothenate in coenzyme A. Okay, so with these particular enzymes, coenzymes and prosthetic groups, they have peculiar structures that helps them in these processes. So The pyruvate dehydrogenase complex consists of three distinct enzymes. The pyruvate dehydrogenase dihydrolipoyl transacetylase, that is E2, and then E3, which is dihydrolipoyl dehydrogenase. Alright, so these three enzymes bind together in stable and non-covalent aggregates, and each enzyme is present in multiple copies with each aggregate. So the number of copies of each enzyme and the actual size of the complex varies among organisms. So when you take the pyruvate dehydrogenase complex from E. coli, the E2, which is the dihydrolipoyl transacetylase, it forms the core of the cluster to which the other enzymes are attached. So E2 has about 24 copies and contains bound lipids, and E1, that is the pyruvate dehydrogenase, contains 24 copies and contains bound TPP, and E3 has to do with 12 copies, which is bound contains bound FAD. So the overall reaction catalyzed by pyruvate dehydrogenase complex, realize this what is being involved over here. From pyruvate to acetyl CoA, you have your coenzyme A, NAD plus, TPP, and lipids and FAD coming into the reaction, and finally you have NAD plus being produced, whilst pyruvate is being converted to acetyl CoA. It's an irreversible reaction and requires substantial action of 3 different enzymes and 5 different coenzymes, like I said. Alright. So aerobic oxidation of glucose, that includes the oxidation of pyruvate, contains or provides energy in the brain. So thiamine deficiency, that's vitamin B1. With patients like that, they wouldn't be able to oxidize pyruvate normally. The disease is characterized by the loss of neural function due to thiamine deficiency, and habitual consumption of large amounts of alcohol can lead to thiamine deficiency also. The beriberi is caused by a deficiency of thiamine, which affects the muscles, heart, nerves, and digestive system. It literally means I can't sing that leaves, and it reflects the crippling effect it has on its victims, and a common part of the developing world where rice is the main food. You have this condition, an abnormal function of pH, PDH, and the TCA cycle, impaired activity of the pyruvate dehydrogenase can result from mutations in the gene encoding any one of the 3 subunits of the enzyme, and deficiency of pyruvate dehydrogenase activity is associated with several metabolic acidosis due to high circulating levels of pyruvate and lactate, with fatigue and hypotonia due to insufficient ATP production. So energy production is impaired in patients with pyruvate dehydrogenase deficiency, especially in neural cells, which are normally dependent on constant supply of glucose as a fuel source. The therapy in PDH deficient persons involves ketogenic diet. That is about 80% fats, 20% protein, and carbohydrates, which creates conditions that where the brain utilizes ketone bodies as an alternative fuel source. So remember the 8 steps of the citric acid cycle. The first, step 1, is irreversible. Step 3 is irreversible. Step 4 is irreversible. and 4 out of the 8 steps are oxidation reactions. Energy is being conserved by electron transfer to FAD or NAD plus, forming FADH2 or NADH2. And all these reactions takes place in the mitochondria. With the TCA cycle, pyruvate, actually acetate from glycolysis, is degraded to CO2, some ATP being produced, more NADH is made, and then the NADH goes on to make more ATP in electron transport chain and oxidative phosphorylation. Let's, like, for example, stage 4, step 4, where there's oxidation of alpha nucleotide to succinyl alcohol A in carbon dioxide. You realize that there's a second oxidative carboxylation step, and the enzyme is the enzyme alpha ketoglutarate dehydrogenase complex is nearly identical to pyruvate dehydrogenase structurally and mechanistically, as there are 5 coenzymes used: TPP, CoA, SH, lipoic acid, FAD plus, and NAD plus, and FAD. So you have to remember that of the pyruvate dehydrogenase to remember that too. So with ATP and GTP, they're rapidly interconverted through the action of nucleoside diphosphate kinase. So GTP is added on to ADP, and there you get GDP plus ATP, which is a reversible reaction, and standard gives free energy, or delta G naught is 0. Alright, so the last thing we'll try and remember has to do with the intermediates of for biosynthesis. The TCA cycle provides several of these, like the alpha ketoglutarate is transaminated to make glutamate, which can be used to make purine nucleotide like arginine and proline, and succinyl-CoA can be used to make porphyrins, and fumarate and oxaloacetate can be used to make several amino acids and also pyrimidine nucleotides. Alright, so we'll continue later.